the male reproductive structure of a flowering plant is called as the stamens. The anther and the filament together they constitute the stamen. So the tapetum, it actually provides nourishment to the pollen grains. An anther, it is bi-lobed. So it is also called as theca. Hello everyone, a warm welcome to today's session on sexual reproduction in flowering plants. So aren't we lucky to be living in a world filled with flowering plants? It is so nice to be gazing at the flowers, the diversity in the color of the petals as well as the scent of the flowers. Flowers keep our mind calm, isn't it? So flowers are used daily in our lives, be it for religious purposes, be it for decorative purposes, and also for other aesthetic events. So from where do we get all these flowers? In spite of the flowering plants having a short lifespan, it is because of the sexual reproduction that takes place in the flowering plants. The flowers produce seeds and the seeds can be sowed in the soil so that a next generation or set of plants can be produced. This is how the life cycle of a flower continues. So before moving on to studying what are the events that occurs in the sexual reproduction of flowering plants, first we need to know about the morphology or the structure of a flower. So we will study or brush up with what are the different parts of a flower. So here I have a simple diagram of a hibiscus flower. So in the hibiscus flower, you know that, or in general in any flower, you know that the flower has 4 whorls. So you have studied about this in your first PUC as well. So the outer whorl, it is the sepal. So the outer whorl is the sepal and it is also called as the calyx. Then you have the inner whorl. The outer whorl is the sepal, which is called as the calyx. Then you have the inner whorl, which is the petal. Or you can also call it as corolla. The petal or the corolla. So inner to the petal, we have one more whorl which is very important for reproduction, which is the male reproductive part of the flower, that is called as the stamens. It is called as the stamens. And inner to the stamens, you have one more reproductive part of the flower which is called as the gynoecium. So the gynoecium is the female reproductive part of the flower. So in spite of flower having all these structures, it is the male reproductive part, that is the stamens, and the female reproductive part, that is the gynoecium or the pistil, that are mainly involved in the reproduction events. Apart from that, the petals also play a very important role in reproduction. Have you ever wondered why is it so? It is because the petals, they are very colorful and they are scented. Most of the petals of a flower, they are scented. So this color and the scent of the flower, it attracts the insect pollinators. So when the insect pollinators come near the flower, they sit on the flower to feed on the nectar of the flower. The pollen grains will stick onto their body, either their antenna or to their legs. And when they visit another flower for the food purpose, again the pollen grains will fall on the stigma of that particular flower and pollination will occur. Therefore, reproduction process or the fertilization events will occur, and finally you will get the seeds which can be stored and which can be sowed whenever needed to produce the next life cycle or generation of the plants. So this is about the structure of the flower. To repeat, the flower, it is made up of 4 important parts, that is the sepal, which is also called as the calyx. Then you have the petal, which is also called as the corolla. Then you have the stamen. So the stamen is made up of the anther and the filament, which we'll be studying in detail later. And the gynoecium, it is the female reproductive part of the flower, which is made up of the stigma, the style, and the ovary. So moving on, we'll study in detail about all these. So apart from this, there is also one more part of the flower, which is called as the receptacle. It is called as the receptacle, or you can also call it as the thalamus. So this receptacle, it is the term that is used nowadays, but earlier it was called as thalamus. So this thalamus, it acts as a stage for the other parts of the flower. So if you can observe this region, all the other 4 parts of the flower are present or bedded on the thalamus. So be it the gynoecium, be it the anther, the sepal, and the petal, everything is placed or it arises from this particular region, which is the thalamus of the flower. So we'll move on to study about each of these reproductive structures one by one. Especially we'll be studying about the gynoecium as well as the stamen. So we'll move on to the most important part of this session, that is, we'll study what are the events involved in the sexual reproduction of a flowering plant. So if a flower has to reproduce, there are certain events that take place, or if it has to fertilize, there are certain events that takes place. So the events are divided into 3 steps. So the first one, it is called as the pre-fertilization event. The second one, it is called as the double fertilization event. And the third one, it is the post-fertilization event. So I'll tell you here a little detail about all these. In detail, we'll study in the next coming sessions. So in the pre-fertilization events, we will study about what are the structures that help in or that are involved in bringing about the pre-fertilization event. And in the pre-fertilization, we have the male reproductive structures and the female reproductive structures. So in the male reproductive structure, how the pollen grains are formed, and in the female reproductive structure, how the embryo sac or the female gametophyte is formed. In double fertilization, so after the pre-fertilization events, once the pollen grains are transferred to the stigma of another flower at that time or of the same flower, what happens is the pollen grains will germinate, they will produce the pollen tubes, and into the pollen tubes the male gametes will move, the 2 male gametes, and they will enter in through the micropylar end of the ovary, and there they will deposit the male gametes inside. So the 2 male gametes, one of it will fuse with the egg cell and the other will fuse with the polar nucleus. Since 2 fertilization is occurring here, that is, 1 male gamete will fuse with the egg cell and the other will fuse with the polar nucleus, it is called as double fertilization. So once when double fertilization occurs, the next process, that is the setting of the seeds and all, will take place. So that comes under the post-fertilization event. Post means after. So after double fertilization, you have the post-fertilization events. So all these events we'll be studying one by one in detail in the coming sessions. In this session, we'll concentrate on the pre-fertilization events, wherein we'll only focus on the pre-fertilization structures that help in bringing about the pre-fertilization events. We'll mainly focus on the male reproductive structure of a flowering plant. So moving further, we'll study about the pre-fertilization structures. So if the pre-fertilization events has to occur in the reproductive phase of a plant, certain pre-fertilization structures are involved. Only if you know in detail what are the pre-fertilization structures, then only you can understand how the events will occur in them. So let's learn about the pre-fertilization structures first. So the male reproductive structure of a flowering plant, Remember, earlier I told you the male reproductive structure of a flowering plant is called as the stamens. So here the stamens are involved. Then you have the microsporangium. When you take a transverse section or a cross section of a stamen, inside you can see the microsporangium, and inner to the microsporangium there are the pollen sacs that are being present. So inside the pollen sac again you will find the pollen grains. So what it means, if the pre-fertilization events has to occur, then all these structures has to be present in a flowering plant. So here we are concentrating on the male reproductive structure, which is the stamen. Inside the stamen, when you take a transverse section or a cross section, you will find the microsporangium, and inside the microsporangium in the pollen sac, you will find the pollen grains. So each of these structures we'll start studying in detail. So here stamens, the stamens, it is actually made up of two important parts. So it is made up of the anther and the filament. So in other words, you can say that the anther and the filament together they constitute the stamen. So if in a male reproductive structure, if the anther and the filament both are present, then you call it as the stamen. So you can see here, you have the anther that is being present and you have the filament. So you have the anther and the filament. Also in this diagram, you can see the anthers shedding the pollen grains. So this we'll study in detail in the coming sessions later on. So you have the anther and the filament. So the anther and the filament together you call it as the stamen. So we'll study the structure of the anther and the structure of the filament. So the anther, it is present at the tip of the filament. You can see here the filament has two poles. So one end of the filament is attached to the anther and the other end will be attached to the thalamus or the receptacle. Why? Because I told you previously, all the parts of the flower, they occur from the thalamus. So the thalamus is the one which holds all the parts of a flower. So one end is connected to the anther and the other end of the filament is connected to the receptacle or the thalamus. So when you see the filament, it is quite long and it is sleeky or slender. So that is the structure of the filament. It is long and it is slender, and you can also call it as a stalk. Why it is a stalk? A stalk is something which holds another thing, right? So here the filament is holding the anther in its place. That is why the filament, it is a long and slender stalk. Next we have the anther. So at the tip of the filament, you have the anther being present. The anther is bilobed. Bi means two. Lobes, it is a loop-like structure. When you see, you can see one lobe here and the second lobe here. So two lobes are there. That is why it is called as bilobed. And one more thing is that you can also call the anther. So the stamen consists of two parts, that is the filament and the anthers. So first we'll concentrate on the structure of the filament. Filament, as I told you, it is quite long and it is slender. And when you see the anther, it is present at the tip of the filament and it is a bilobed structure and it is attached to the thalamus or the flower. In the previous slide, you can see that the stalk of the anther or the filament, it is attached to the petal as well as the thalamus. So the anther is present at the tip of the filament. It is bilobed structure and it is attached to the thalamus and the petal of the flower. So the proximal end, that is the free end, the one end of the filament is attached to the anther and the other end of the filament is attached to the thalamus. And also you can see that in a flower in different species of flowers, say for example, you keep 10 different species of flowers on a table and you start to observe the anthers. Some flowers may have only 2 anthers, some may have 4, some may have 6, 8. Like this, the number of anthers present in a species of flower keeps on varying. And not just the number of anthers, even the length of the anther also varies. Why does the length of the anther vary? It is because of the length of the filament is different in each species of flower. Some flowers may have 2 short filaments and anther. The other flowers may have 2 longer filaments and 2 shorter filaments. Like this, it keeps on varying. That is why the anthers, they are different. When you see the flower, the stamen or the anthers are different. Why? Because in one flower, the anthers may be just 2. In other flower, the anthers may be 4 or 6, like that. And the length of the filament of the anther also varies. So next, I told you an anther, it is bi-lobed. So it is also called as theca. So each lobe of an anther, it has 2 theca. So here again you have the filament. You have the filament in the anther. Then you have the anther. So together you call it as the stamen. It is bi-lobed, having 2 lobes. It is bi-lobed. And since it is bi-lobed or it has 2 lobes, it is also called as dithecous. Instead of writing lobes, you can also write it as it has 2 theca. So that is why, since it has 2 theca, you call it as dithecous. And each theca is separated by a longitudinal groove. So you can see a small depression here. This is called as the groove. So each theca or each lobe of the anther, it is separated by a longitudinal groove. And when you take a transverse section of an anther, you can see that it is tetragonal. Tetragonal, it has 4 corners or 4 sides. That is why it is called as tetragonal. So when you take a transverse section of an anther, so here in the diagram you can see clearly when you take a transverse section of an anther, it has 4 corners. That is why it is called as tetragonal. That is why it is called as tetragonal. And in each of the corner, you have the microsporangia. So in the locules, so this is the locule. So here you can see clearly, this is the locule. So in the locule, at the corners, you have the microsporangia. So totally, there are about 4 microsporangia. So you have 4 microsporangia. As I told you, 1 microsporangia here, 1 here, 1 here, and 1 here. So totally, you have 4 microsporangia. And inside the microsporangia, you have the pollen sac. So inside the microsporangia, you have the pollen sacs. So pollen sac. Sac is something to hold a material, or to fill something, you call it as a sac. Something that holds another material or something, you call it as a sac. So pollen sac, obviously it will hold the pollens inside it. So inside the pollen sac, you can see here small structures which are spherical. These are the pollen or the pollen grains. The pollen or the pollen grains. So when you take a transverse section of the anther, you can see that the anther has 4 corners or 4 corners. That is why it is called as tetragonal. It is tetragonal, and each corner, it has 1 microsporangia. Therefore, the anther has a total of 4 microsporangia. And inside the microsporangia, you have the pollen sac, and inside the pollen sac, you will have the pollen grains. Inside the pollen sac, you will have the pollen grains. So this is about the structure of the stamen. That is the male reproductive part of a flower. So moving further, we'll study in detail. I told you, when you take a transverse section of the anther, inside you have the microsporangium. So let us study about the structure of the microsporangium, because it is very important to understand the structure of the microsporangium if you need to learn the pre-fertilization events. So the TS of the microsporangium, it actually appears to be circular. So when you see a microsporangium, it is circular. Almost the outline of the microsporangium, it is circular and it has 4 corners. That is why it is tetragonal. Tetra means 4 and 4 corners. That is why it is called as tetragonal. And when you observe this particular transverse section of the microsporangia under the microscope, you can get a weave like this. This is the enlarged weave. So, what are the structures that are present in the microsporangia? We will study. So, the microsporangia, it is surrounded by or it is covered by 4 wall layers. That is the epidermis, which is the outer wall layer. So, epidermis, epi means outer, dermis or derma means covering or skin. So, epidermis is the outer covering of the microsporangium. Then, inner to the epidermis, we have the endothecium. And inner to the endothecium, we have a set of middle layers. And inner to the middle layers, we have the tapetum. So, we'll study in detail what each of these layers or the covering, what function they perform in the microsporangium. So, as I told you, microsporangium is surrounded by 4 wall layers. The outer wall layer, it is the epidermis. The outer wall layer is the epidermis. Inner to the epidermis, you have the endothecium. So, endo means inside, inner. Epi means outer, outside. So, we have the endothecium. Then, inner to the endothecium, we have the middle layers. So, the middle layers, and inner to the middle layer, we have the tapetum. And apart from that, we also have the microspore mother cell. So, this is the MMC. It is the microspore mother cell. Microspore mother cell. So, when you look at the anther in a larger weave, that is the microsporangia, when you take a cross section of the anther, you find the microsporangium. When you observe the microsporangium under the microscope, you can see that it has 4 corners. That is why it is called as tetragonal, and it is covered by 4 wall layers. What are the wall layers? The outer wall layers, that is the epidermis. The inner wall layer, it is the endothecium. Inner to the endothecium, you have the middle layers, and inner to the middle layers, you have the tapetum. So, what are the functions of these layers? So, the endothecium, the epidermis and the middle layer, they protect the anther, because inside the anther is the one the pollen grains are produced. So, they are the most important reproductive structure, right? So, if reproduction has to occur, the pollen grains has to be safeguarded. So, the main function of the epidermis, the endothecium and the middle layers is to protect the anther. And the function of the tapetum, so the epidermis, the endothecium and the middle layer, they perform the function of protection to the anthers, and also they help in the dehiscence of the pollen grains. So, when the anther matures and when the pollen grains are about to be released outside, the anthers have to split open. So, if they have to split open, then the middle layers, the epidermis and the endothecium, they have to break open. So, the epidermis, the endothecium and the middle layer, they not only protect the anther, but they also help in dehiscence of the anther so that the pollen grains can come out and the further fertilization events can occur. So, I also told you there is one more layer that is called as the tapetum. So, the tapetum, it actually provides nourishment to the pollen grains. So the pollen grains that are present in the pollen sacs. So where are the pollen sacs present? They are present inside the microsporangium. So those pollen grains, they need to get nutrition for the survival or for the nourishment, right? So the function of the tapetum, it is to provide nourishment or nutrition to the developing pollen grains inside the microsporangium or inside the pollen sac. So the cells of the tapetum, when you observe, they are very dense or thick. Why? Because it is filled with thick or dense cytoplasm, and it is made up of more than one nucleus. That is, it is multinucleate. Multi is more than one. Nucleate, nucleus. So it is dense, filled with cytoplasm, and it is made up of multinucleate. So you can see here, this is how a microsporangium occurs under the microscope. So when you take a cross section or a transverse section of the anther and observe, this is how the microsporangium occurs. So you can see the outer layer. I'll repeat again. You can see the outer layer, which is the epidermis. Then you have the inner layer, which is the endothecium. Endothecium. Inner to the endothecium, you have the middle layers. The middle layers, inner to the middle layer, you have the tapetum. So this tapetum, it provides nourishment to whatever pollen grains are present inside. So you can see here the pollen grains being present inside the pollen sac. So this tapetum provides nourishment to the developing pollen grains. So this was about the structure of the microsporangium or the anther. So anther, it is made up of a filament, or in general you can call it as the stamen, which is made up of the anther and the filament. Then you have, when you take a cross section of the anther, you can find the microsporangium. The microsporangium is covered by 4 layers, and the first 3 layers, they perform the function of protection to the pollen grains, and also they help in dehiscence of the anther so that the further pollination process and all that can occur. And you have the tapetum layer. So the tapetum layer, it provides nourishment to the pollen grains. So now we'll move on to the last structure of the male reproductive organ in a flowering plant, which is the pollen grains. So pollen grains are very important in order for the fertilization or the reproduction process to occur. So the pollen grains, they are also called as the male gametophyte. So I'll tell you why they are called as the male gametophyte. So male is, obviously it is because they are produced by the male part of the flower, that is the stamens. So in the stamen, the anther is the one which produces the pollen grains. And gametophyte, why? Because when you take a section of the pollen grain and observe, inside you can see 2 gametes. So if this is the pollen grain, you can see 2 gametes inside the, this is the male gamete. So you can see the male gamete. That is why it is called as gametophyte. So gamete, it is the male reproductive structure in a pollen grain. The 2 gametes, male gametes, phyte or phyta means plant. They belong to a plant. That is why they are called as the male gametophyte. So the pollen grain is also called as the male gametophyte, and they are spherical. So when you observe a pollen grain, they are either circular or spherical. And so you can see all the pollen grains, they are almost spherical. So all the pollen grains, they are spherical. They are almost spherical. So the pollen grains are spherical, and they have a measurement, that is their diameter is about 50 micrometers. So usually the pollen grains have a diameter of about 50 micrometers. So why do we say it is a diameter? Because it is circular, right? That is why the measurement is in diameter. So it has 50 micrometer diameter. And when you see the cytoplasm of the pollen grain, it is surrounded by the plasma membrane. So the pollen grain, inside the pollen grain, you have the cytoplasm. In the cytoplasm, you have the vegetative cells and the 2 generative cells, or you can call it as the male gametes. And also inside these you have the nucleus being present. So the cytoplasm of the pollen grain, it is surrounded by a plasma membrane, and apart from that, pollen grains, it can be stored for years, right? You can store the pollen grains for years, and whenever that plant is extinct or something, from wherever you have stored it, you can just take the pollen grains and you can bring about fertilization again, and you can bring back the species of that particular flowering plant. And don't think that all the pollen grains will have the same lifespan. So the viability or the lifespan of the pollen grain from one flower to other, from one species of flower to other, it varies. So what are the reasons why it varies is because maybe because of the humidity or the temperature, they keep on varying. So the viability of the pollen grain, they vary greatly, and it mainly depends on the humidity as well as the temperature. And also the pollen grains are of different forms. So there is a diversity of pollen grains. So you can see here the wall of the pollen grains, they are so beautifully, it is of different forms. Why is it? It is because of the decorations that are present, or the design of the exine, which is the outer layer of the pollen grain. That is why you can see different forms of pollen grains. In spite of the shape being spherical or circular, they look different, and they have a diameter of about 50 micrometer. So moving on, we'll study the structure of the pollen grain. So when you see a pollen grain, I told you it is spherical. So the pollen grain is spherical and it is covered by two layers. So what are the layers? The first layer is the exine, which is the outer layer, and the second layer is the intine. So exine has a very good property which protects the pollen grain to a longer duration. So that is why I told you for years together we can preserve the pollen grains. So we have to thank the exine. Because of this exine, the pollen grains can be stored for a longer duration. I'll tell you why it is so. So I told you pollen grain has two layers. One is the exine and the other is the intine. So the outer layer of the pollen grain, it is the exine, and the inner layer, it is the intine. Again, exine, ex, it is outside. Intine, in, it is inside. So that is why it is called, the outer layer is called as the exine, the inner layer is called as the intine. So exine, it is hard outer layer or the hard outer covering. So since it is hard, that is why you can preserve the pollen grains for a longer duration. So the exine, apart from that, when you see the exine on the pollen grain, you can see pores like this. The pore will not be present only on one region. It will be present wherever possible in different regions. So inside this pore, it is called as the germ pore. So the pollen grain, the exine of the pollen grain, can you see here? You can also see spines. Why all this is present is because it will increase the rigidity, or it will increase the toughness of the pollen grain or the toughness of the exine. Therefore, pollen grains will have a longer lifespan. So that is why spines are being present, and also you have germ pores. So why germ pore is needed? Because to facilitate easy germination. So that is, germination is easy through the germ pore. That is why germ pore is present. And also not just that, on the germ pore you have a special material that is being present. It is called as sporopollenin. So I'll just mark here. It is called as sporopollenin. So throughout the exine you have the sporopollenin being present. Sporopollenin is present covering the exine throughout. But wherever this germ pore is there, say for example, the pollen grain has a germ pore here or a opening or aperture here. But here sporopollenin is not present. It is absent. It is absent here. It is not present. So it is only present where the germ pores are not there. So germ pores are very useful, which helps in germination, or it helps the pollen tube to come out of the pollen grain. So for that, germ pores help. And apart from that, wherever the germ pores are not present, on the egg sign, there is a special material that is present. It is called as the sporopollenin. So this sporopollenin, it is a very tough compound or a chemical compound. It is a very tough chemical compound. So the sporopollenin, it helps the pollen grain to withstand high temperature and also withstand high acidic as well as strong acidic and alkaline conditions. So that is why it is very, very important. So next we'll move on to the inner layer, that is intine. So the intine of the pollen grain, it is very thin. So intine can be thin. Why? Because most of the protection work will be done by the exine itself. So that is why intine, it is thin layer and it is thin and it is continuous. So why is intine continuous? Because in intine, these germ pores are not present. The germ pores are present only on the exine, but in the intine, the germ pores are absent. That is why it is a continuous layer. At a stretch, it surrounds the pollen grain. But the exine, since it has apertures in the form of germ pores here and there, that is why it is not continuous. So you can observe here. So this is the exine. You can see here the pollen grain, exine. Here and all there is no covering. Exine is not covered. Why? Because it is not continuous. Because germ pores are present. That is why it is not continuous. But in the case of intine, it is continuous. It is at a stretch. It covers the pollen grain. Why? Because there is no germ pores being present. So this is about the exine and the intine. So we'll study about inside the exine and the intine. There are certain structures being present inside the pollen grains. So what are they? I told you earlier itself, the pollen grain has a vegetative cell and a generative cell. So when you see a mature pollen grain, it has two cells. So it has two cells. One is the vegetative cell and the other one is the generative cell. So we'll study about them one by one. So in this diagram, you can see a pollen grain. So pollen grains, they undergo mitotic division to produce pollen tetrads. They undergo first mitotic division and they form two cells. In the second mitotic division, they form the four cells. So this in detail in the coming sessions we'll study when we study about the pre-fertilization events. So it is a pollen tetrad now. I'm just giving you a small introduction here. In the coming session, when we study about the pre-fertilization events, we'll study in detail about how the pollen grain formation occurs. So here you have the pollen grain. They undergo the first mitotic division and they undergo the second mitotic division. Second mitotic division to form a pollen tetrad. Why is it called a tetrad? You can see there are four pollen. So here inside one grain itself, there are four pollens being present. That is why it is a tetrad. So when you observe one pollen grain, say for example, here when you observe one pollen grain, you can see inside you have the vegetative cell. You have the vegetative cell and you have the generative cell. So we have the vegetative cell and the generative cell. So a mature pollen grain, it is made up of two cells. One is the vegetative cell and the other one is the generative cell. So the vegetative cell, it is bigger. So can you see here? It is bigger. When you compare the vegetative cell with the generative cell, the vegetative cell is bigger. The generative cell is smaller. The vegetative cell contains abundance of food material. So it is a food reserve. It contains abundance of food material. So the vegetative cell, it is bigger it has abundant food reserve, and it has a large irregularly shaped nucleus. Why irregularly shaped nucleus? Because the nucleus is neither round or any circular or something like that or spherical. It has no shape. So you can see here the nucleus doesn't have a particular shape. That is why it is called as irregularly shaped nucleus. So the vegetative cell, it is bigger when you compare it with the generative cell. It has a dense cytoplasm, and in the cytoplasm it has a lot of food materials being present. Why food material is being present? Because it has to nourish the generative cell or the male gametes. That is why it is being present. And when you see the generative cell, it is small and it floats in the cytoplasm of the vegetative cell. So the generative cell, it is small and it floats in the cytoplasm of the vegetative cell, and it is spindle shaped. It is spindle shaped. So can you see here? It is spindle shaped. Spindle shaped, and also it is made up of dense cytoplasm. That is, it is filled with cytoplasm, thick. It is very thick. It is filled with dense cytoplasm and a nucleus. So as you can see here, the vegetative cell, it is larger than the generative cell. It is filled with cytoplasm, which is abundant in food reserve or food materials. It is a food reserve, and it has a irregularly shaped nucleus. And the generative cell, it is very small. It floats in the cytoplasm of the vegetative cell. It is somewhat spindle shaped, and it consists of a dense cytoplasm as well as a nucleus. Here the nucleus is proper. It is not irregular. It has a proper shape. But in the case of vegetative cell, the nucleus is irregular. So this was about the structure of the pollen grain. So I told you pollen grains are very, very important to bring about the fertilization events. If the seeds have to set in a flower, then pollen grains are very important. But the pollen grains also have certain disadvantages to humans as well as animals. So first we'll study what are the advantages of pollen grains, and then we'll move on to what are the disadvantages of the pollen grains. So the pollen grains, I told you, they are rich in nutrients. Why are they rich in nutrients? Because they have a sucrose source in them, and also the vegetative cell of the pollen grain, it is filled with food materials, different sorts of enzymes, sugars and all that. So that is why they are rich in nutrients, and hence what different companies, what they do is they collect this pollen grains, they crush it, they make it into a powder, and then they make tablets out of it. So since that tablet is rich in nutrients, it provides a lot of energy to whoever consumes it. So that is why these athletes and all, they consume this pollen grain supplements. So pollen grains, they are rich in nutrient, and hence pollen tablets are being used as a food supplement by the athletic people, or whoever is in sports, they prefer this pollen grain tablet. So this is one of the best advantages of the pollen grain. So moving on to the disadvantage of pollen grain. So pollen grain, it actually causes allergies. So most of you might be experiencing when there is during this rainy season or the winter season, when there is heavy winds and all that, you know that pollen grains, they travel in wind very easily because they are very light, right? So at that time what happens, the air will be filled with different sorts of pollen grains. So the pollen grains of some of the plant species, they actually cause harm to human beings. They bring about or they cause respiratory disorders such as asthma, bronchitis, or sometimes you'll have watery eyes, you'll have a running nose. So all that is because of the pollen grains. So pollen grains, though they have an advantage in the sense they are used as food supplements because they are rich in nutrients, right? Apart from that, there are also disadvantages. How the pollen grains causes the pollen grains, especially of some of the plant species, they cause respiratory diseases in the human beings. So the best example is the Parthenium. So you might have seen, everybody have seen fields which are filled with Parthenium nowadays. Even in the roadside, everywhere you have this Parthenium plant, right? It is also called as the carrot grass. So this Parthenium plant, it is not native to India. It came into India from another country. So when Indians, when we were importing wheat from another country, along with the wheat seeds or the wheat grains, these Parthenium seeds also entered India, and that is how now India, wherever you go, be it in a wasteland, be it in the agriculture field or in the roadsides, you find this Parthenium plant. So especially this Parthenium plant is the one, the pollen grains of the Parthenium plant causes a lot of respiratory disorders, such as you have various chronic or severe respiratory disorders such as asthma, bronchitis. Apart from that, you get various allergies related to the eyes, the skin, and all that. So this is the disadvantage of pollen grains. So here, hope you have understood about what the different structures of the male reproductive organ of a plant are. So you studied about the stamen, you studied about the microsporangium, and you studied about the structure of the pollen grain, and you also came to know what are the advantages as well as the disadvantages of the pollen grain. So I hope the session went well and you understood whatever I told you. So in the next session, we will study about the female reproductive structure, that is the pistil or the gynoecium, and in the female reproductive structure, we'll study about the megasporangia or the ovule, and we'll study about the female gametophyte, which is called as the embryo sac. So this female reproductive structure also comes under the prefertilization structures. So hope you understood this session. Thank you.